Hola, muy buenos días. Bienvenidos a los seminarios del Instituto de Biología de la Universidad Nacional Autónoma de México. Hoy, martes 1 de septiembre de 2020. Cada martes, para los que no están muy familiarizados, cada martes a esta misma hora, hora de la Ciudad de México, llevamos a cabo los seminarios de nuestro instituto. Como verán, hoy estamos personas diferentes a cargo del seminario, porque hoy es el primer día en el que el nuevo comité, como les mencionó la directora del instituto la semana pasada, el nuevo comité está integrado por la doctora Alejandra Moreno, por el doctor Ulises Rosas y por un servidor Lázaro Guevara. Quiero aprovechar para agradecer nuevamente al comité pasado que estuvo a cargo de Camille Tron, Marcelo Pase e Ivonne Garzón, que hicieron un trabajo extraordinario, sobre todo en esta transición debido a la pandemia, para pasar todos los seminarios a una forma virtual. Entonces, de verdad quiero reconocer mucho el trabajo de ellos y también quisiera explicar brevemente cómo va a ser la dinámica de hoy en adelante, ya que tratamos de que sea un poco diferente el sistema de seminarios. Va a ser una transición relativamente lenta y vamos a estar recibiendo anfitriones del instituto, es decir, estamos invitando nosotros como coordinadores a gente del instituto para que ellos nos propongan a investigadores. Hoy es un ejemplo de ello. Y vamos a tener esta figura de anfitriones que el día de hoy tenemos al doctor Ulises Rosas, como verán en unos minutos. Después también queremos invitar a algunos investigadores técnicos académicos del Instituto de Biología, es decir, gente de casa. Cada mes tendremos a un invitado. El primer invitado va a ser el doctor Fernando Álvarez, a quien vamos a escuchar dentro de 15 días. Today we have the pleasure to have Dr. Rachel Meyer with us. And el anfitrión es Ulises Rosas. Rachel, thank you very much again for being here with us, for preparing your talk. Sin más, voy a dejar al doctor Ulises Rosas para que haga la presentación de su invitada. Muchas gracias.
Muchas gracias, Lázaro. Pues hoy tengo el placer de presentar a la doctora Rachel Meyer. Rachel es una experta en botánica, etnobotánica, genómica de la domesticación y DNA ambiental. Rachel habla inglés, chino, alemán y entiende español. Es por eso que, de acuerdo con Rachel, esta presentación la vamos a hacer en español y espero que ella entienda. Rachel obtuvo su licenciatura en Plant Biology de la University of Washington en el 2006. En el 2012 obtuvo del City University of New York y del New York Botanical Garden con su asesora, la doctora Amy Lead, una experta en evolución del desarrollo. A partir de ahí, ella ha tenido diferentes trabajos. Del 2012 al 15 fue becaria postdoctoral de la NSF Plant Genome Research Program. En 2012, ella fundó la empresa Shoots and Root Beaters, que es una empresa de educación en ciencias botánicas. Ha sido becaria en política de la ciencia y tecnología de la American Association for the Advancement of Science, National Science Foundation. Recientemente ha sido la directora ejecutiva de la University of California Conservation Genomics Consortium, que es de la University of California, Los Ángeles. Y actualmente, desde 2019, es Assistant Professor in Ecology and Evolutionary Biology de la Universidad de California en la Universidad. Rachel tiene experiencia en la acción de financiamientos para investigación, tiene una gran cantidad de artículos publicados, tiene 31 en total, más 2 en revisión actualmente. Ha publicado en las revistas muy prestigiosas como PLOS Genetics, Environmental DNA, G3, BMC Plant Biology, F1000, Nature Plants, Nature Genetics, eLife, Molecular Biology and Evolution. Ha sido invitada a presentar 23 seminarios y ha impartido otros 25 seminarios por contribución en eventos académicos. Y me gustaría mencionar que Rachel es una persona muy talentosa desde el punto de vista artístico y algunos de sus trabajos han sido portadas de Nature Genetics, PLOS Biology, e inclusive la ilustración que ven hoy en el seminario, en la página de YouTube del seminario de hoy, pues es una ilustración que ella Así que, sin más preámbulos, welcome, Rachel, to the Institute of Biology seminar series. So the microphone is yours.
Thank you, Ulysses. And it's so good to see you again. It was really great to do a postdoc with you back in New York. And nice to meet all of your wonderful colleagues. I look forward to a fun conversation today. And thank you for putting up with my seminar for the next 45 minutes or 40 minutes. All right, I'm going to try to share my screen. Great, I'm assuming this is working fine. So, like Ulysses said, my background is in plants mainly, and in the last couple of years, it's been a really fun roller coaster ride diving into broader biodiversity inventory and involving the public and trying to explore what different types of systems level biodiversity work we can do here in California. So thank you for allowing me this opportunity to present the work that we're doing to you. So my talk is called Systems Level Biodiversity Sampling by Community Scientists and Environmental DNA, a framework for California conservation. I'm sure that people here are familiar with biodiversity hotspots. Most of Mexico is a biodiversity hotspot, and in the United States, we only have a handful of them. California is one of the 3 or so biodiversity hotspots in the United States. Of course, the California floristic province that is a biodiversity hotspot extends into Mexico and has more than 10 times the diversity of most of the central and northeastern United States and Canada. So we have an incredible amount of biodiversity to protect while also expecting about 50 million people to live in our state. We also are one of the agricultural centers for the United States. So a lot of the land is dedicated to agriculture and natural resource extraction. And climate change is already threatening half of the wildlife and biodiversity hotspots worldwide. In California, this is no different. We have tremendous challenges, and the places where a lot of our wildlife live are also the places where most of us like to live as well. Here's an image of one of our local endemic species. This is the giant sequoia, which you cannot easily fit in a photograph. And the recent wildfires in California have actually burnt a lot of the habitat where sequoias live. The large sequoias that are really towering, they fortunately have survived, and a lot of the understory has not, and that's actually part of what makes a successful old growth forest. But fire suppression in the area has led to tremendous biodiversity change and a lot of loss of these old growth understory species that would normally be part of this habitat that is fire adapted. So that's just one illustration. You may have heard about it in the news, and of course there's many places around the world that are dealing with wildfires in this season where there's just tremendous challenges with making habitat suitable for humans, which is usually suppressing fire unless we employ more diverse practices, considering Native American practices of burning the landscape that had worked for thousands of years, but since Europeans came to California, we haven't done that so much. Another example of a California biodiversity protected species is the California condor, which is a huge, beautiful bird that was almost extinct and the populations have been fortunately rebounding in very select locations in California. Recently, one of the challenges that we're dealing with is with very limited habitat where the condor lives, an arsonist just last week set on fire the condor sanctuary in Big Sur, California. And fortunately, this little baby condor is still alive, but it's very difficult to monitor that population right now. So we're also dealing with not just climate change and more people living here, but human-induced big problems. And so a limited understanding of and appreciation of biodiversity is critical to change. So the condor, after, this is not my work, but this is, I love the story of the return of the California condor. You can actually see right now we don't have very much genetic diversity in the condor, and we have a reference genome for it, but there's ancient DNA that we can get from museum specimens that people have published beautiful studies to show that there used to be much more genetic diversity of the condor. And you can use museum specimens from Washington to Mexico that actually help us understand the haplotype diversity, haplogroup diversity. This type of ancient DNA from museum specimens I'm going to talk about because it actually leads to why we're interested in environmental DNA. So a museum specimen, that DNA is quite degraded. You can kind of picture it like Beth Shapiro, who I work with closely at UC Santa Cruz, calls it the confetti after a big party. Say you're at a parade and that confetti washes down, into the gutters on the street and gets mixed with all sorts of other stuff. And what we're doing is taking those little pieces and putting them back together, trying to map them to a reference genome. Often there's a lot of bacterial DNA and non-target DNA. So from that museum specimen of a California condor, you might have 20% of the DNA you extract belong to condor, and the rest of it belongs to a whole bunch of other species. Maybe the human that touched that, that stuffed the animal to prepare that specimen, for example. So here at UC Santa Cruz, we have a pretty awesome Paleogenomics Lab where some of my colleagues have extracted DNA from nearly million-year-old specimens. Most of these are like horse bones, 740,000 years old. And so the expertise of some of the people in this lab and at other UC campuses got together and decided to start the UC Conservation Genomics Consortium back in 2016 and apply their ancient DNA wisdom to environmental DNA, and also brought in reference genomes and population genomics, landscape genomics, and software that would be much more applicable to California biodiversity managers. So we're using DNA barcoding and DNA metabarcoding for environmental DNA. Like we said, for ancient DNA, we're dealing with a soil sample or a water sample where there's loose DNA that's been shed by organisms into the environment. It could be skin cells that have been sloughed off. It could be fecal material. It could be leaves that are decaying slowly through time. And all of these different organisms that are shedding DNA, that DNA is rapidly breaking into smaller and smaller pieces as it's being exposed to UV light, to oxygen, to water, to acid, to all of the different elements in the environment. So we frequently rely on short fragments of DNA, usually DNA barcodes, to detect a species or a taxon and try to match the piece of DNA that we have to a specimen, usually a vouchered specimen in a museum. So these are just from Wikipedia, some of the more common DNA barcodes that are used. And what we do is we amplify these in using very conserved priming sites. These priming sites here where you don't see very much variability, and target a hypervariable region that usually varies from species to species. And these barcodes have been developed since the early 1980s. When you use next generation sequencing, you can basically take your environmental sample and your soil sample or your sediment sample or water sample and use your barcoding primers to do metabarcoding. So you amplify everything that those primers could bind to, and then you sequence all of it. And with informatics pipelines, you match all of those sequences to all of the species in the reference database that you can get and try to make an inventory of biodiversity from that one sample. So in a sample, we typically get about 1,000 different taxa. And we know that we haven't sequenced everything on Earth yet, right? We only have about, well, from the tests that we've done in California using the Global Biodiversity Information Facility, about 89% of the families in California have at least one reference DNA sequence that's been published, but only half of the species in California that are known, of course we can't even talk about the unknown unknowns, have some DNA available. So we're doing a match to the closest match possible. And it's good for tallying or counting biodiversity, but it's not necessarily going to be perfect. In fact, at the species level, there are still plenty of errors because we haven't sequenced most of the species yet. But here's an example from a study that I did on the Russian River. There's 172 sites on this x-axis and taxa on the y-axis. And what you get is this amazing kind of fingerprint, like a barcode in itself, of what biodiversity looked like in the Russian River. What are the taxa that are always there and kind of ubiquitous, and what are the taxa that you expect them to be very rare? And you could actually use this little stamp to track how biodiversity is changing through time and through space. And that's what makes this really exciting to build new biodiversity baselines. Environmental DNA, we thought, if we apply this to California, there's an opportunity to solve really big, complex challenges and to address a lot of different fields of science, social science, earth science, climatology, to genetics and environmental justice with environmental DNA. And we can ask questions about restoration, about how to reseed biodiversity after, say, a wildfire comes through. We can ask about resilience and recovery, and we can try to understand what are some of the refugia that we have that help store biodiversity and help reseed it to other areas in California. So this is our name of our program, Cali DNA, for California Environmental DNA. And some really beautiful artwork that my friend Maya did when we got started. This, I think, really helped us get volunteers. Our goals were to characterize the biodiversity of ecological systems as a whole, to collect data over the state from the same point in time, which no single lab can do. So that required us to activate the public and collect data and help that public not just be data collectors, which a lot of community science or citizen science programs do, but to interpret results with us, because nobody is an expert of all this biodiversity. And the community is more of an expert on their local biodiversity, their local ecology, than a scientist coming in who's not from there. So we really want to work with people whose eyes are always watching their local area, and when they see something change, they see something that wasn't there before, they say, oh, well, this used to be here, but it's not here anymore. That type of biodiversity change is very useful for us to align with environmental DNA. And we would really like to increase fluency and interest in biodiversity, genomics and genetics, and obviously conservation. So we have a website, ucedna.com. You can go online to register and sign up for BioBlitzes, where we go out as groups, or especially in COVID, where we send you a kit and hope that you stay safe going out on your own. You can collect samples, analyze, and then you send this back to us. We analyze them in a lab, and often have students working on those projects. And then together we explore the results. We put the results immediately online and invite people to look at them and give us some feedback. And we've had several volunteers join the lab as interns, co-author manuscripts, And it's a really exciting relationship that we're building. And we've started working with NASA Jet Propulsion Laboratory and some of the national labs and community colleges and high schools. And we're trying to really branch out what we're doing. And we're not just based in California anymore. That was the start. But we actually have projects that are in different states and even internationally. So we, for example, have been working with a really awesome professor, Lida Castro, in Colombia to survey the beach myofauna. So I hope that this little cartoon works, just a vignette of the field experience. You have a little kit that has meters, you have a phone app, and you have your gloves and tubes, and you just collect some environmental samples, and everything's pre-barcoded for you. And then you can send that back to the lab. With or without abiotic data, we also rely heavily on satellite data. And then we also ask you to take photographs and put them on iNaturalist so that we can use those for ground truthing. We pay a lot of attention to metadata. Environmental DNA is like a whole new field. So permit holders, like land managers who would give you a permit to do your research, often don't know how to give permission for environmental DNA. They're like, oh, well, it's soil. It's nothing living, except it's giving you information about something living. So we're also trying to build relationships with people in the state to help recognize how to document and receive permission for these types of samples to be collected, what that communication should look like, how do they want results communicated to them. And there's a lot of really interesting bioethics questions that I'm not going to go into here with environmental DNA, but we know that because it's that confetti type of thing, that if I'm collecting environmental DNA from one location where I've received permission in a river, I'm actually getting information from upstream in the river where maybe I don't have permission. So there's a whole bunch of issues that can make this a very interesting space to be in on the policy side. And then so what we do, we have a dynamic web platform. We use Kobo Toolbox. It's an international free like phone app that you can use to fill out a form. You don't even need to have a cell phone service. It's very secure. It's partially funded by the Gates Foundation so that you could like be in the middle of, you know, Oman in a rural desert and be able to collect survey data. So it's mainly used by social scientists. And so this way, when policy changes, when the criteria change, oh, now we need you to document the proximity to a road or the depth of your sample, we can add that to our forms and still have the information connected to our collections. And then our laboratory methods are all online. We try to connect with different biodiversity groups, such as the Global Biodiversity Information Facility and the Global Biotic Interactions database. And we have software that some really brilliant postdocs and graduate students made for doing the multi-locus metabarcoding informatics called AnaKappa and RanaKappa. So quickly, our little overview of our methods is we have these samples that have been stored in a minus 80-degree freezer for 100 years. They're going to be there. And as part of the UCLA collections, we take 3 samples from each site, or the volunteer would take 3 samples from each site as biological replicates. We usually pool them into a single sample. We just take a little aliquot from each, and these the biological replicates remain as reference collections that you can actually check out internationally. If you're doing a project on desert soils, we could lend you some soil and you could find all the diatoms or tardigrades or whatever you want from there. We extract DNA, we do our multi-locus metabarcoding. We added a primer, so now we're also looking at vertebrate 12S. And then we do MySeq or NextSeq sequencing, and then use our bioinformatic pipelines to do filtering for quality control and taxon assignment. And here's, you know, some of the fun that we get to have in the field. I forgot there was volume on this. So it's a lot of fun. And so from those samples from the Russian River, I'll just use that as another example. Here's a tree of all of the biodiversity, the taxonomic biodiversity that we found. We used the synthetic, the open tree of life to make this phylogeny. And so it's a synthetic tree. And from all of this biodiversity, we actually find things that are unexpected sometimes. So in the Russian River in California, we found giraffe DNA. Like, this is a very sensitive method. We get so many different taxa, and we also can, with metabarcoding, even if most of the actual DNA in the sample is bacteria, you can still detect really rare things. And the giraffe is an example of this. So in one of the samples from the Russian River, there was giraffe DNA, and I couldn't believe it. And it turns out that the creek, Mark West Creek, where we had collected that sample, is right next to a safari. I had no idea this was in California, but these giraffes are shedding DNA into the creek about a mile from where we collected the sample. We also have this really cool opportunity when we have lots of different sites to not just characterize holistic biodiversity in a place, but we can actually make these co-occurrence networks. So in this example, we have lots of eucalyptus from a handful of sites, and that's enough to actually associate, like when you detect eucalyptus, what bacteria are you detecting at the same time? What fungi are you detecting at the same time? And we can kind of think of these as candidate holobiomes. They're in a network together, they co-occur, and so maybe this is telling us something about their ecological function or their trophic relationships. So it's pretty exciting that once we generate all of this data, we can start to infer ecological roles. One more example of this is the plant Ludwigia. It's a primrose. It's aquatic, and it sort of grows along the riverbanks. We detect it in a lot of different sites. That's what you can see here. And so we can actually try to understand what are the associated taxa with Ludwigia that help it become so destructive as an invasive species here. So it starts to grow, bacteria colonize on it, form a biofilm, diatoms come to live there, algae come to live there, and then you have this whole little microbiome of stuff that acidifies the area. And so you have terrible blooms and eutrophication, and then the Ludwigia die, and that exacerbates more different bacteria, further acidifying the habitat. So we can actually see with the samples that have Ludwigia all of these different organisms that co-occur with them, and that can give us an idea of why they have this process that this cascade of biodiversity change that they cause. It's not just them, it's all of the diatoms and algae and fungi that are associated with them. So we rely heavily on this website, so all the data that we put online, because we want to be able to see how frequently we observe something. So now we have about 1,000 sites with DNA results, and I'm still just wrapping my head around what we can do when we have 1,000 sites with biodiversity measurements. What we can ask is what is generally common and what is uncommon, where we really need more observations to be able to say, make that co-occurrence network, or to understand what are just generally associated habitats and species. So here are 2 invasive species in California, Festuca, which is a type of grass, we can see we track it all over the place, so it's very common where we've sent volunteers. But tree of heaven, which is a terrible invasive, I'm sure in Mexico as well, and that we for some reason only have in one site so far. So we really need people to get out there and sample near these invasive species so that we can understand how they're completely changing the entire habitat. So here's our website, and I told you that we do these little bio blitzes where we have groups go out at the same time and we try to survey a whole area. But we also send people out, say, in the same weekend on their own. We say, okay, go to 40 sites on the coast. So what's wonderful about community science is you can activate people. And so in one weekend, we can get an inventory of all of California's 1,000 miles of coastline. Sorry, I should be talking in kilometers. And then also we work in groups where we work with courses, undergraduate courses, and we develop different teaching modules, which has been really exciting. And these are some of the undergrads in my lab that we just started at UC Santa Cruz. And so far they're happy. We'll see how long that lasts. And we have these really fun group bio blitzes where we've had actually like over 100 people out at a time, all age levels. They go to vernal pools. These are pools that in Mediterranean systems exist where in the winter time, you have water from the rainy season, and then in the summertime, that water dries up. So the pool only exists for part of the year, and you have these hard clay soils. So you get evolution adaptations to just drop your seeds very locally because you are adapted to a certain amount of water at a certain time of the year. And if you were to be at the edge of that pool, you would be less adapted. So you get these beautiful rings of different species, these beautiful flowers. It's just an incredible sight. There used to be vernal pools all over California, and we've destroyed 95% of their habitat. So there's not very many left, but we can have volunteers go out and collect lots of samples of the vernal pools, try to understand how different the different levels of the pools are, and what are some of the species that maybe through traditional surveys without eDNA we haven't been able to track. So we're here comparing the bottom of the pool to the edge of the pool and the upland adjacent areas of the pool. Here's an example of Calusa grass, Neostaphyia colosana. It's endemic to vernal pool systems, and it's only existing in a couple of pools now. What we could see with eDNA is actually before the grass emerges, we would get an eDNA signal. So you can almost use it to predict when there's going to be that grass coming up that year. In California, we often see like 3 years, you don't see a grass, and then 1 year you suddenly see the grass. So we have such different years variation in our rainy seasons from El Niños and La Niñas that it's just very sporadic. So eDNA has been really helpful to characterize what's in the seedbed and what's emerging. So here's some of our different primers: plant ITS, CO1, fungal ITS. Sorry, the S got cut off for some reason. 18S and 16S. 16S is for bacteria, 18S is for general eukaryotes. CO1 is for mainly animals like invertebrates. So we can compare different years where volunteers have gone out to collect samples. And here we see this is alpha diversity. So the overall number of species in these 5 vernal pools that we surveyed for invertebrates was higher in 2018, which was a dry year, than in 2017, which was a wet year. But in bacteria, we see the opposite. In the wet year, we see more species richness than in the dry year. And the fungal diversity mirrors the invertebrate diversity, as does the plant diversity. So it's really fun to be able to see these general biodiversity patterns, looking at alpha diversity, beta diversity, and zeta diversity. And the networks have also been interesting to look at from year to year. So in the wet year, 2017, we see a plant and algae network that looks very different from a dry year. And the placement of invasive species in the network, in the wet year, they weren't incorporated into the network, but in the dry year, the invasive species were incorporated into the network. So this has been a very interesting new emerging area of study for us, and I would love to talk with people that do these type of analyses at UNAM and maybe form some collaborations. I'm going to talk briefly about work from Meishi Lin, who is a graduate student at UCLA. She's interested in doing landscape community analysis and asking what are the biodiversity patterns. So maybe from biotic biodiversity, we have our eDNA details of what samples have what species, and we can do these beta diversity plots, taking different sites and seeing how they cluster, how similar the communities are. But we can also relate this to abiotic data and ask, how does the environment help us predict or explain the beta diversity patterns that we see? And so we're using a method called gradient forest to do this. It's a statistical modeling approach of the composition of the different biodiversity at different sites using a variety of environmental predictors. So we use mostly things that you can get from Google Earth Engine, and on our website, we actually had an undergraduate build a Jupyter notebook so that you could download all of this data for your samples if you just upload an Excel file or a table of geolocations. So we have location, human impact, bioclim, soil properties, topology, some of the Sentinel-2 satellite data that gives us an idea of photosynthetic activity and local weather and climate, sand and clay and other organic material properties in the soil. We take all of these different data, and then we take everything that's autocorrelated and we remove anything that's autocorrelated at an R squared at above 0.8. And then we just run gradient forest. For this data set, she had 915 families in 278 sites across California that were collected by volunteers, and 33 environmental variables from those abiotic matrices. And by associating them, she was able to find the top variables that predicted biodiversity across all of California. And this was elevation, which is comforting to know because for over 100 years, we've known that elevation is a major driver of biodiversity turnover. The amount of sand, vegetation coverage, and the mean temperature of the wettest quarter, as well as total nitrogen. But our overall predictive power is still quite low. So we need to have a better understanding of what the temporal footprint is of environmental DNA. How much, we don't expect that the environmental DNA signature that you get on a certain day is best aligned with the climate of that day. We have to figure out what is the best window of time to measure from the environment to align with that environmental DNA signal. And that window might vary depending on if you're looking at bacteria or insects or plants or algae. And that's just because the life cycles are very different for these things, whether you're measuring something that was there and it's degrading or it's currently living in that system, like soil bacteria. We expect very different results. So we have some grant proposals and we'll see what happens. But still, she was able to make a prediction with the gradient forest method that was much better than by chance, which is comforting. And project across all of California, not just the 278 sites that were measured, the upper left map, which is a biodiversity community turnover map of California. And we can compare that map that's using environmental DNA and abiotic data. We can compare that to all of the other maps that have taken decades to build, from the USDA, from the Environmental Protection Agency. We have all these different maps of California that are based on other data, and they have nuances, they have differences in them. So we can compare our map to those maps and ask, where are the borders? Where does the habitat really change the most? And through time, through seasons, and from year to year, do these gradients change? And of course, we also have, with these lots of sites, we have networks that we can build. And we can start to ask questions of, well, how are invasive grasses perhaps getting an edge, having high fitness in the California environment? Maybe it's because they have these associations with really interesting other species. Just to highlight some of the other work that we've done, after the wildfires in Southern California around Los Angeles and the Santa Monica Mountains, we started looking at how biodiversity was changing after wildfire. And so right now we're currently optimizing how to collect, how to sample DNA from these heavily burned soils that have a lot of inhibitors and kind of nasty chemistry in them. And then we're trying to take all of our data and run it through some really neat models about post-fire reassembly and associate them with some of the NASA predictions of environmental gradients. That is a rooster crowing. I hope that you can't hear it, but it's bothering me. I apologize. We are babysitting a rooster for our friends right now. Anyways, so there's also some fun projects. This is Dr. Tiara Moore, who is now a postdoc at the University of Washington and the Nature Conservancy. She looked at lagoons and systems that, these systems where eutrophication happens, you get this ulva macroalgae bloom in the lagoon. And so she could actually not just do the, so she brought some volunteers in and they did the environmental DNA sampling, but then not just metabarcoding, she took the samples and used them for a geochip analysis to look at stress genes and could actually associate specific bacteria with pectinases and all sorts of other enzymes that were causing this algal, this eutrophication from the decaying algal matter. So really exciting to be able to see people apply the community science collections to their research. And here's a network that she built of all of the different organisms that are involved in that eutrophication process. So it's tremendously diverse. And finally, I'm going to just highlight another project we're really excited about, where we're starting to work directly with managers. This is from my hometown. I'm from a place called Palm Desert. It's a small town with lots of oases and golf courses and not much to do if you're a kid. But the Boy Scouts of America dumped a whole bunch of crayfish into this beautiful oasis in the 1950s as a thank you gift, and also introduced tilapia to this oasis, thinking what they were going to do was an ecological experiment. You'd have crayfish and tilapia and they would regulate each other's populations. That did not happen successfully. The tilapia were in the deeper part of the oasis and the crayfish were in the more shallow part of the oasis. And so they didn't actually regulate each other. They just ate everything and really destroyed this habitat so that not very much could live there. But, and here's an example. If you look into the oasis, there's like 30,000 crayfish. You can, I mean, they were everywhere, and about 4,000 tilapia. So the oasis managers were set on removing the crayfish, but they wanted an environmental DNA method to help ensure that they were able to sensitively track if the crayfish were going to come back. So we sampled sediment and water. We actually collected water in these 1 liter bags and hung them passively so that they could be filtered without us doing very much work. And then we extracted DNA from the filter. It was fun. We got to kayak on the oases. This is a volunteer from the desert, Rachel Stark, who actually got married on the oasis. So she is doing some biology with us. And we also had crayfish and tilapia in these different tanks. So we had 3 tanks of crayfish, 3 tanks of crayfish and tilapia, 3 tanks of tilapia only, and 3 tanks that were blank. And from all of these samples, we generated potential bioindicators. So immediately after we collected the samples and we went to the lab, the oasis shut down. It put up a big fence and said, we're closed, we're going to apply pesticides, we're going to eradicate the crayfish. So that was about a year ago. They drained the oasis and they poisoned it every month for a full year. And they also used bait traps to see if crayfish would come back. And we've now done 3 sampling points to try to look for these associated microbial species that were associated with crayfish before the crayfish were removed. We're trying to track them to see if they come back. So here is Simone Pond. This is the oasis that had the crayfish and tilapia. And here is another oasis that was pristine. There were never any crayfish or tilapia there. And we see a lot of taxa that are very simple statistical analyses that are always in the invaded pond and never in the pristine pond. And some of these were also in the tank experiments. So that gives us higher confidence that they're actually associated with crayfish. And some of them actually come up in the literature as being part of the gut microfauna or microflora of the crayfish, the same species. And here's where it's been so nice to have this website database with other sites in California where I can ask the question of, oh, is my detection of Talaromyces, this supposed crayfish-specific microbe, is that something that I am likely to just find anywhere in California? Maybe it's just a component of the soil or something associated with other plants, or other insects, other types of organisms. And I can see that in 1/3 of my sites, I get Talaromyces. And this has been so important for us to help us build these bioindicator taxa and guide the oasis of if it's likely that the crayfish are actually gone. So really nice to have this sort of baseline data set. And also we're able to build really cool kind of community resources, I think. We have some undergraduates and recent graduates at UC Santa Cruz in the lab who are monitoring the return of biodiversity after these invasive species have been removed. And so they can look up species like this eyelash fungus, which is really cool looking. And they can ask, okay, is it very common in California? And we find it's only found in 8 sites in California, and most of those are in our oases. And we can use GBIF, the Global Biodiversity Information Facility, to also confirm that it has been found in California before, which gives us some confidence that we are, in the right ballpark type of taxon. So that's just a little story of some of the work that we're doing in the lab. And we're really trying to scale up and build partnerships. And again, it's a huge challenge. It's a new area for us. We've only been working in this for a couple of years now. So really excited to talk with you about environmental DNA and learn maybe what's happening in Mexico. So thank you.
Thanks a lot, Rachel. It was a very interesting talk on the interface between state-of-the-art genomics, citizen science, biodiversity, and science communication. We have a few questions from the public. The first one comes from Noemi Chavez Castañeda, who is an expert on ornithology. And she wonders, she knows that you are a plant specialist, but as you can see, Rachel manages a lot of things from plant biology, genomics, environmental genomics. So she's asking if you can comment a bit more about the effects of habitat loss on condor populations currently in California.
This is a really good question for Ollie Ryder at the San Diego Zoo, who is a close colleague. Right now, I think what I see in the news is the fires are the biggest habitat loss for the condors. Just a couple years ago, they also lost a significant amount of habitat, and their populations are still very small. So I'm curious to hear your expertise on this and your opinion of what might help us broaden their habitat, and could environmental DNA help us understand maybe a safe place to introduce them, maybe a place where there's no parasites or pathogens that are known to affect them. It could be something that helps us do restoration. I don't have a good understanding of why they can live in one place and not another. The Pinnacles National Park has been a great place for condors, and it's apparently very easy to see them there. So if you come visit, we'll go.
Thanks, Rachel. Also, Laura Margarita Marquez Valdemar, who is our sequencing facility manager at the Institute of Biology. She's asking, she understood that you get the 3 samples from every site, and then you pull them together, right? So she's asking why not to analyze them separately. And she's asking what technique you are using for extracting DNA, and she's congratulating you for your work.
Thank you, Lara. It's nice to meet you. We in the past, we had a very limited budget, so we were pooling the samples together because indexing was expensive. But now we make our own indices, and we use the dual unique indices. So we are increasingly sequencing each biological replicate separately, and increasingly sequencing technical replicates separately. Each PCR that you do is very stochastic. So you can get a different biodiversity profile from one or the other. From each sample, we do 3 PCRs for every metabarcode. But to understand how unstable that alpha diversity statistic is, it's really nice to have the 3 separate PCRs done. Beta diversity is extremely stable. And then for sequencing, we usually try to get 50,000 reads for every sample for every metabarcode. And so we've been trying to move over to like the NextSeq or the NovaSeq when we have short barcodes that are under 300 base pairs. I've seen some really cool work using like PacBio, but we haven't tried it ourselves yet.
Okay, thank you, Rachel.
And then for DNA extraction, it's the QIAGEN Power Soil Kit, which works great to remove inhibitors. Sorry, Ulysses.
Good. While we wait for a couple of more questions, I have to say that Rachel is making a big effort not to miss this seminar, because she was indirectly affected by the current fires in California. And I have to say that I was a postdoc colleague of her back in New York with Michael Purganen. And back then, Rachel was working primarily on domestication. So I have a question for you, Rachel. What can we learn about environmental DNA regarding domestication, which I'm sure you have heard about?
Oh my gosh. I'm so excited that you asked this. So we actually do ancient environmental DNA analyses of human middens from like 8,000 years ago in California. There's caves. It's the trash that people have had for thousands of years. And we go through the trash, and we find signals of maybe some of the plants that people ate. For the animals people ate, and domesticated and fished for, we often have bones or some sort of fossil evidence. But for plants, sometimes we don't have that evidence. So we're actually trying to use ancient environmental DNA to help complement archaeological records and patterns of plants that are just bigger around archaeological sites, to propose that in California, domestication of plants happened. there's so many Native American groups here that still heavily use California native species for foods, and there's no reason that they are not domesticated and shouldn't be counted as domesticated, especially with a broader view of of what domestication is, that it's an ongoing process. So one of the clear ways is just to help use ancient environmental DNA to fill in the records about who was using what when, and how we moved plants around the state and maybe around the country or the continent. And of course, for domestication, the changes in the plant chemistry and maybe some of the plant fungal, plant microbial relationships, you can see that with environmental DNA. With oak trees, we can even see that the same species of oaks, but from different parts of California, maybe from a drier area, a higher elevation area, a wetter area, they have significant different associations with nematodes and rotifers and all sorts of different organisms. And we can detect that through environmental DNA in the nearby soil and in the leaves. That's the rooster again. Ulysses, it's so good to talk plants again. Thank you. I've been really missing it.
Well, we have a huge amount of biodiversity here in Mexico, and I hope you come to visit us here at the Institute of Biology and get to talk to the colleagues, because I'm sure there is a lot of avenues for collaboration here in Mexico. And following up on that question, can you calculate somehow the footprint of human activities on biodiversity using your eDNA?
I think so. It works better for terrestrial systems than for aquatic systems so far. We have human impact, like human footprint data sets for the state, and they're very coarse. So the alignment with our eDNA profiles is not perfect. But if we can get more high resolution data, maybe when you're collecting a sample, maybe when you fill in your phone app, you can add some information about the human impact in the area, and maybe that'll help. I'm really curious to explore that. Would love to talk more about that and understanding urban impacts on biodiversity.
And I'm sorry, but I have one more question. And so it's regarding change, and I can see that more is more. I guess scanning more biodiversity gives you more information, which I guess gives you better predictions. But can we, or can you identify taxa that you think are better predictors for climate change in ecosystems?
Yes, yeah. So with the gradient forest example from Meishi Lin that I showed you, for each family that we had, so of the 915 families, each family, you see what are the top environmental predictors. So we actually know which are the families that are the most environmentally sensitive, which ones are the most temperature sensitive or rainfall sensitive. And what's really scary, and I didn't present this in a slide today, but maybe I should have, but I'll say it now, is that often we think you're either biotically sensitive or abiotically sensitive. Like you're dependent on a relationship with some other local species or with the rainfall, for example. What we're seeing is the most environmentally sensitive species or families are also the ones that have the strongest biotic relationships. They're the most networked species. So if they drop out because of climate change, what happens to the biotic network? Does it fall apart? Mm-hmm. It's scary.
Yeah, that's very, very interesting. We have a question from Hernan Vázquez Miranda, who is an expert in our institute on ornithology, genomics, and bioinformatics. And he's asking, what would be informatics infrastructure to carry out eDNA study, time, RAM requirements, data storage, and so on? So what do we need for us to do that, to do what you do?
Hernan, first, I have to compliment you on your Darwin drawing of the coral of life. That's one of my favorite sketches of his. And yeah, so for the informatics, it's really nice to be able to parallelize the blasting step. So if you have a server where you can parallelize blast, that's great. Everything else, most of it you can actually do on a desktop or laptop computer. It's really just the taxon assignment and then bootstrapping your alignments that takes a lot of computer time and power. And I'd be really happy to work with you. We have a containerized version of our software, Anacapa, that has, you know, as a container, it's got version control for all of the different other accessory software, Python versions and stuff like that that you need. But there's so many new software for environmental DNA coming out. I would be really excited to see more that's available on the cloud, because we have a lot of colleges that don't have servers that that they can use, and it'd be really nice to just upload your data and get your results. So yeah, if you hear of anything, or to just see what's out there, what your preferences are, and really curious what different people are doing with environmental DNA.
Thank you, Rachel. We have a question from Alberto Hernandez Horta. He's saying it would be interesting to know if there is a change on eDNA due to the pandemic. And excellent work. So have you thought about it?
Alberto, yes, totally. This is the largest change in human impact that we've ever seen. So suddenly since the Industrial Revolution, of course. What's really cool about environmental DNA is we can collect small samples and just put them in the freezer, right? We can store them. Even if we don't have funding, we can store them, and we have a time capsule. That way we can go back and we can track at this point in time. It's really hard to know where that change is going to happen. With the eDNA change due to the pandemic, with biodiversity change due to the pandemic, we can go back to where we had samples collected, and we can recollect from those sites and see if we see some difference. But having those multiple time points is really important, and you never know where you're going to want to focus on some natural disaster that happened, or some major change, maybe a factory closed, right? And people are trying to look at environmental RNA and wastewater to detect coronavirus. So they've actually talked to us about doing that, but we've said, no, we don't want to get involved. It's a little over my head.
Well, thanks a lot, Rachel. So it seems like there are no more questions. And all what I have to say is that thanks a lot, Rachel, for agreeing to give this seminar, this virtual seminar. I promise I'll get Rachel to come to Mexico so Rachel can talk to the people at the Institute of Biology, and hopefully also teach a course on bioinformatics and eDNA to the students and academics at the Institute of Biology. With all of this, thanks, Rachel, again. Rachel's email is on the screen at the moment, and Rachel is a very open person for communication, for collaboration, so please get in touch with her if you're interested on the methods, bioinformatics, and collaborations that you might establish with Rachel. As I said at the beginning, Rachel is starting a new lab in UC Santa Cruz, so not only for collaborations, but also for students. Rachel is a very enthusiastic person who is always welcome to receive internship students or graduate students or even
yeah. Anyway.
Or even just to talk, go for a coffee and talk science. Rachel is a person to talk about a whole range of topics, including science, domestication, arts, and so on. So thank you, Rachel. And I just have to say some finishing words.
Thank you so much.
I want to say thanks again to Camille Trong, Yvonne Garzon, and Marcelo Pash for being the previous coordinators of the seminar. They did a wonderful job doing the transition from presential seminars to virtual seminars, and we are very thankful for doing that. And now we have new coordinators who are Lázaro Guevara, Alejandra Moreno, and myself, Ulises Rosas. And we have this new system, relatively new system, in which we invite the community of the Instituto de Biología to function as a host of our speakers. So in this occasion, I am the host of Rachel. So in the future, hopefully any of you part of the community of Institute of Biology can be the host of a speaker that you fancy to listen or fancy to share their contributions to the science community. So with all of this, thanks a lot, Rachel, again. Thanks a lot for being with us in this seminar series. And see you later. See you next week. Next week, I have to say, we have the seminar of Dr. Carlos Martorell from Facultad de Ciencias, and the host is Dr. Andrea Martínez Ballester from Jardín Botánico Instituto de Biología. So we wait for you next week, same time, same channel. Thanks a lot, and see you soon.
Muchas gracias.